So this is just a video about transcription translation that I found on YouTube, but I'm just recording it so that I can narrate the key points that you guys need to understand and help clarify some of the concepts in the video.
This animation demonstrates how the digital information encoded within DNA is used to direct protein synthesis. This is a DNA double helix containing the digital code which directs the cell in all aspects of operation.
So we are inside the nucleus right now, which is where our DNA is kept.
And here we see a protein complex called an RNA polymerase traveling down the DNA strand. As it moves down the strand, it carefully unwinds the DNA, preparing it for transcription. Inside the polymerase, we see a single-stranded copy of the original instructions.
So, in order to get a copy of the original instructions, we are reading it off the template strand. So this is the template strand, and this is my coding strand. And that makes sense because if you look at it, my coding strand and my mRNA that's being made are identical.
Being assembled as individual bases are positioned and added to the growing strand. A stop code marks.
So this is my actual mRNA strand that's being made by the polymerase.
The end of the protein specification, at which point this copy, known as a messenger RNA transcript, exits the polymerase and heads towards a two-part chemical manufacturing machine called the ribosome.
So before it can actually reach the ribosome, we know that it has to leave the nucleus. And before it leaves the nucleus, it has to undergo RNA processing, which is splicing out our introns so that only exons are leaving the nucleus, and also adding that 5' cap and the poly A tail. All those things have to happen before it can actually leave and get to the ribosome.
While the messenger RNA moves towards the ribosome, transfer RNA molecules attach to specific amino acids in preparation for assembly. As the messenger RNA transcript passes through the ribosome, the process of translation begins.
So transfer RNA is just what we call in class tRNA. So those are the same things. And it's reading these codons on my mRNA and matching it up with this right here, which is the anticodon. And soon you're going to see at the top of this tRNA a little red puzzle piece, and they keep connecting together, and those are amino acids.
Using the instructions encoded on the messenger RNA as a
amino acid right here being attached to this chain, and when we have a chain of amino acids, we call it a polypeptide.
Template, the transfer RNA molecules align specific sequences of bases to corresponding amino acids, creating a protein chain.
Okay, so you can see that this is your polypeptide, your protein chain that's being assembled by the ribosome. Now, all this other stuff that I'm going to fast forward through, you don't need to worry about. We're not going to cover it in this class, but I just want you to see that eventually we end up with this. A folded protein that's ready to go out into the cell or throughout the body and complete its specific function. So these are the different elements of protein folding that we talked about in action. So hopefully that helps give a good visualization of what transcription translation look like in the cell. And if you still have any more questions, make sure that you come see me in office hours.